Our first keynote of the date is Dr. Marvin Hausman, Chief Medical Officer at Pilz Biosciences Corporation. Dr. Marvin Hausman received his medical doctorate degree from the New York University School of Medicine. He's an immunologist and board-certified urological surgeon. And has over 30 years of drug research and development experience with various pharmaceutical companies, including Bristol Myers International, Mead Johnson Pharmaceutical, Squibb Medical Research. He's an accomplished executive with domestic and international experience, who is also on the alumni board of governors of NUI Grossman School of Medicine. Dr. Hausman, the floor is yours.
Thank you. It's a pleasure to be here. Thank you for the introduction, Christina. I had the pleasure of listening to Peter van der Heyden, and I think it's nice to follow him because he mentioned certain things. Well, one was the stigma of the industry. The other word he mentioned was evidence-based. And what I'd like to do in my presentation is to bring the industry into the 21st century of medical knowledge. And I think if we do that, we will have a future around psilocybin. So my topic is strategy for making psilocybin clinical trials more successful. May I have the next slide, please? So what we want to do is we want to define a glossary. We have to define the playing field. Where are we with respect to drug development? And psilocybin is our drug. So we have phases of clinical trials. Preclinical is the proof of concept animal study. This is very important. You produce a 95% pure drug for the regulatory agencies, but then you don't even know if it's going to work. So you need an animal model that's well validated. Once you have that, and you have a proof of efficacy, you go into a phase, what we call a phase 0, initial human trial, and that tells you how the drug's processed. How does it affect the body? Small number of patients, 10 to 15 subjects. Once you do that, you then go into phase 1. Find the best dose of the new drug with the fewest side effects. Small number again, N to 15 to 30 subjects. That's called, both of them have called ADME, absorption, distribution, metabolism, excretion. And then you go to phase 2, which is the additional safety study. But now you evaluate in a specific condition. And I'm going to show you specific conditions that are being worked on by well-known companies in the psilocybin and psychedelic space. Phase 3, tough study, double-blind placebo-controlled trial. Compare your drug to the standard of care drug. N, large number, more than 100 more patients. And once your drug is approved, you can do post-marketing studies. The FDA loves post-marketing studies. Several hundred or thousands of patients. You can look at outcomes research. Is what you promoted really occurring? Next slide, please. So what are the products we're going to develop? We could develop drugs, which this is where my lecture is, a medicine or other substance which has a physiological effect when ingested or otherwise introduced into the body. Then there are nutrients. These are chemicals we eat every day, stay healthy. These are chemical compounds found in food that are used by the body to function properly and maintain good health. Medical foods. Now, these are foods that are specially formulated and intended for dietary management of a disease that has distinct nutritional needs that cannot be met by normal diet alone. A medical food, for example, to ensure if you have an intestinal disorder or you've undergone surgery and you have limited intake of food, medical food such as Ensure give you all the proteins and all the nutrition, all the minerals that you need. Next slide, please. Now, in our world, we're looking at psilocybin, and the mushroom, there are many different mushrooms in the world that we want to research, and some of them are down in Mexico, some are being cultivated. The largest series known is the Psilocybin mexicana, but the one that's being cultivated most for the industry is Psilocybin cubensis. And before you, you have the substances that are in Psilocybin cubensis. Psilocybin, norpsilocin, psilocin. Just want to touch, and then you have at the bottom other psychedelic derivatives. Psilocybin is not the active form. Once you ingest psilocybin, it becomes psilocin, which is the active form. Next slide, please. So the pharmacology of mushrooms. Mushrooms are the largest kingdom in the world, food-based, 40,000 different species. They could be ingested as a whole natural food. They can be used for individual compounds. We now synthesize, for example, psilocybin, and we can extract it. The future might be in extraction, not in synthetic. Next slide, please. Natural formulations. Nature is perfect. We're all part of the evolutionary process, whether you believe in medicine or God. I don't get into the politics and religion, but we are perfect. We've evolved, and nature is absolutely perfect. And when it creates something like a food, it's been created over thousands and thousands of years. And there's a thing called the entourage effect, that the active compounds inside the food complement each other. That's why they were put there. Vitamin C was put inside an orange or a lemon, not to take it out. It's much more active with the biochemical efficiency of the food that created it. Next thing, food safety, low toxicity, less side effects, non-addictive as compared to psychiatric drugs. Drugs are chemicals. Foods are not. And for example, let's take psilocybin argentevis. If you compare the pure psilocybin with the mushroom extract, say in OCD, obsessive compulsive disorder, you find that the therapy of the mushroom extract is better than the psilocybin alone. And that's a study by Matsushima. And all of these studies that I'm going to quote are available for distribution if you contact me. Next slide, please. We now have to go to the regulatory authorities, whether it's the European EMA or the US FDA or the Canadian Health Organization. Good manufacturing practices have to be adhered to. And that is a system to ensure that the products that you're going to ingest and use are consistently produced and controlled according to quality standards. Early on, when you do your preclinical in the animal, you use non-GMP, which is a batch intended for non-clinical use. However, it has to be greater than 95% purity. You're allowed, under the regulatory agency jurisdiction, less than 5% impurities. When you go to human use, the letter C stands for current. And it reminds manufacturers and the drug companies that they must employ technologies and systems which are up to date in order to comply with FDA regulations. Next slide, please. Now, I want to go through all of the landscape, and then I want to show you how to design the study to meet regulatory authority approval. So let's look at some of the current psychedelic drug landscape. These are well-known companies. Compass Pathways is developing psilocybin in treatment-resistant depression, Mind Medicine, LSD, in anxiety disorders, Zynerga Pharma, cannabinoids, transdermal, they're using a transdermal approach to apply them, Fragile X syndrome, and Pilz Bioscience, psilocybin in autism spectrum disorder. There are general symptoms, and most of these are behavioral anxiety, mood disorders, hyperactivity, intellectual disability, poor communication. How do we integrate these behavioral symptoms into a drug approval process? Next slide, please. This is an example of Zynerga just completed a treatment with Zygel, behavioral treatment outcomes and significance. It was a phase 2 to 3 study. And what I want you to look at here, and it's a complex chart, we can go into this during questions and answers, a treatment value with a P less than 0.05 is statistically significant. If you look at the left side of the chart, you will find that only 1 out of the 4 behavioral scales that were used was statistically positive, with a p less than 0.02, and that was the social avoidance subscale. Now, any improvement came close at 0.05, and Zynerga now is going before the FDA to say to them, well, we have this subset of Fragile X. Can we get the drug approved? And I'll leave it to questions and answers, and we can deal with it once my presentation is completed. Next slide, please. So Compass Pathways has a clinical trial. I think they're in phase 3. It's a synthetic formulation of psilocybin called COMP360. This is a phase 2B. I think they're in phase 3 now in treatment-resistant depression. The objective of this study is the pharmacological effects of psilocybin, a psychoactive substance, with psychological support. And we're going to address that. Next slide, please. So what is the outcome measure that they're using, including this primary psychological support? Well, one is the safety and efficacy of psilocybin in participants with treatment-resistant depression. That's your phase 1, phase 2. You have to have it or you can't go on to phase 3. They're using 3 treatment arms: psilocybin, low, medium, and high. And they're looking at a Montgomery-Åsberg depression rating scale over 12 weeks. The higher the rating, the more severe the disorder. So this rated scale measures depression severity from a score of 0 to a score of 6. They'll have about 10 items and questions with a total possible score of 60. So this is a very subjective behavioral examination. Next slide, please. If you look at another company that's actively involved, they just made an announcement. Mindmen announced its first ever clinical trial combining ecstasy, MDMA, and LSD. It's a phase 1 clinical trial. Of course, LSD, as you well know, is colloquially acid. It's a hallucinogenic drug. And they are dosing LSD from 25 micrograms up to experiential doses of 200 micrograms. The maximal good drug effect was reached at 100 micrograms. And the behavioral test that they're looking at is called ego dissolution. And this is thought to be one of the key therapeutic potentials of the psychedelic experience and psychedelic-assisted therapy process. Next slide, please. So MindMed, similar to what we discussed for primary outcome, because this is going to be very important when you go to the regulatory authorities, the experience of a compromised sense of self, termed ego dilution, is a key feature of the psychedelic experience. Now, this is an experiential approach to getting drug approval. The ego dissolution inventory is a new 8-item self-report scale designed to measure ego dissolution. The clinical evaluation will investigate the specificity of the relationship between the psychedelic compound and ego dissolution. I want you to keep all this in mind as we move forward to create this paradigm. So I wanted to lay the landscape. What is the industry as we see it today? Next slide, please. Now, very important question. Can the study of human behavior be objective? Why do certain behaviors occur? You have to stand back and you have to be able to investigate that behavior in an impartial way. Now, human behavior can be studied scientifically, and I'm going to show you a big breakthrough there, but it is not easy. And let's take an example of a current behavioral well-used study called the Social Avoidance and Distress Scale, SADS. It's a 28-item self-rated scale used to measure various aspects of social anxiety, including distress, discomfort, fear, anxiety, and the avoidance of social situations. Post-traumatic stress disorder, Parkinson's disease, Alzheimer's disease, stress related to cancer, all have these behavioral abnormalities. In fact, there is now theory that suicide is really an excitatory potential, and the brain can't control the energy that's being released between nerves, and that person commits suicide. It's not a genetic abnormality. It's an inflammatory reaction to a current social situation. Next slide, please. This is a very interesting disorder. It's not really a disorder, it's really a spectrum. And when we use the word spectrum, we're saying it's a basket of disorders that we really don't understand the subsets of autism, specific subsets that go into the spectrum. And it represents a huge market, 3.2 billion in 2018. It's forecast to reach 4.6 billion by 2026. We're going into 2021. It's a global disorder, and it's an unmet medical need. It's a developmental disorder that affects communication and behavior. Now, some of the key things that in the DSM-5 to specify this spectrum: difficulty with communication and interaction with other people, restricted interest and repetitive behaviors, symptoms that hurt the person's ability to function properly in school, work, and other areas of the life, and are part of the disorder. Next slide, please. The pathogenesis still remains unknown, except it's a huge unmet medical need. Theories and connections. This is now promoted. Autistic behavior arises from dysfunctions in the midbrain dopaminergic system. I'm going to talk about that. Don't be confused by the word dopaminergic. Elevated blood serotonin system, present in more than 25% of affected children. And here's the conundrum. So we have a central nervous system disorder. How can we explain that when you use antibiotics in an autistic child, the symptoms improve? What's going on? What don't we understand about this problem? Next slide, please. So, serotonin system, just to lay the groundwork, give you a little bit of the landscape. It's called the 5-hydroxytryptamine. And by the way, all of these are proteins. This is a hormone that is a protein. It stabilizes our mood, feelings of well-being, and happiness. Interesting. Serotonin is manufactured in the brain, where it performs its primary functions. However, 90% of the human serotonin supply is found in the digestive tract, the small intestine, and in the human blood platelets, which come from the bone marrow. And also, there's a problem. You cannot measure it in the brain of a patient undergoing drug development. Next slide, please. Dopamine system, similar. Protein, hormone, neurotransmitter. It sends messages between cells. It's inside the brain. It plays an important role in executive function, motor control, motivation, arousal, reinforcement, and reward. However, again, it cannot cross the blood-brain barrier, and you can't measure it in a human being easily in the brain during a drug study. Next slide, please. What are the new frontiers in this disorder that's an unmet medical need? We just discussed, we have to deal with it. Why is it a spectrum? Why is there nervous system function disorder? Why do we have disorders in behavior? And then we have to bring this industry into the new world. What is the new world? Just like I'm speaking to you, there's a language inside the body. Our cells are communicating. That inflammatory language is RNA, ribonucleic acid. And I'm going to explain that as best as I can. There's also another language called the microbiome. Next slide, please. Does nature have the answer? Absolutely. Superman comes into it. Antioxidants, free radicals, food, nature has the answer. Now we have to define how we get to that answer. Next slide, please. So I don't know if most of you know this, and I bet you a lot of you take probiotics. Probiotics are the good guys in your gut, and the food that feeds them is called a prebiotic. What you may not know is that the human body is composed of 37 trillion human cells, but 100 trillion bacteria. So we're really 3-to-1 bacteria. How do we stay alive? How do we walk? We're 70% water. How are we held together? Probiotics maintains the beneficial microorganisms, improve or restore your gut flora. But you have to feed your probiotics, and that's called prebiotics. That's non-digestible food ingredients, such as fibers, and your bacteria loves that, but they don't like the terrible high fructose sugars that corrupt them. But you want to promote the growth of beneficial microorganisms in the intestine. They have to work together synergistically, prebiotics and probiotics. If not, you end up with leaky gut syndrome, a compromised immune syndrome, constipation, pure mental health, increased blood pressure, weight gain, etc. Next slide, please. Now, this is fascinating. Parkinson's disease is in the brain, but the disorder begins in the small intestine. And you have a GI disturbance, and you really examine Parkinson's disease patients. And James Parkinson, the English surgeon who discovered it, most of his patients had constipation. He missed it. And we now know that the microbiome gets corrupted in the small intestine, and it produces the inflammatory molecules that migrate up the vagus nerve into the brain. Is that also occurring in depression related to cancer? Is it occurring in post-traumatic stress disorder? Is it occurring in epilepsy? Is it occurring in Alzheimer's disease? It goes on and on and on. Next slide, please. Epigenetics, very important 21st century term. The study of changes in organisms caused by modification of gene expression rather than alteration of the genetic code itself. Very important concept. Your gene DNA is fixed, but the expression of that gene can be modulated. And we're going to look at that. Next slide, please. So a successful clinical study, and this is where I wanted to lay the foundation and the landscape, involves, this is a Venn diagram of how to design a professional study that would be accepted by the regulatory agencies. On the left, we have behavioral analysis. In the middle, we have ASD, autism spectrum disorder. But we need to bring in these other modalities that I've mentioned that are now in the 21st century of medical studies. The immune system. Think of COVID-19, cytokine storm. People are dying because the immune system is out of whack. The virus is exploding your immune system to create the storm of inflammatory molecules in your lung. That's why you go on ventilators. Gut microbiome. We're going to talk about that also. The probiotics. You are 3-to-1 bacteria, so you better pay attention. And I explained earlier that autistic children are better when you give them antibiotics. Are the microbiota much more important than we've ever realized? Next slide, please. Language of disease, my language, I'm speaking to you, but your cells, this may be Star Wars thought processes, but your cells talk to each other. The bacteria talk to each other. Cancer cells talk to each other. This is a picture of a DNA and an RNA transcript. And what happens is the RNA takes a picture, forms a template of the DNA, and then moves as a messenger into the cell, into the nucleus, moves into the cytoplasm as a messenger. Next slide, please. And so messenger RNA is a single-stranded RNA molecule. It's much smaller than DNA. It's a version of the gene that leaves the cell nucleus and moves to the cytoplasm where proteins are made. An organelle called the ribosome then comes along, lines up along the RNA, reads its base sequence, a language is being created, and uses the genetic code to produce proteins. The language of inflammation is associated with these pro-inflammatory cytokines, which are proteins. We've got to learn that. We have to identify with that. And we have to modulate it if we're going to cure diseases or have an effect on diseases that are unmet medical need. Next slide, please. Cytokines, very important. Inflammation and autism spectrum disorder. Let's look at some evidence that's in the literature, which I think has been overlooked by many scientists who are developing drugs against autistic disorders. One, The white blood cells in autistic people, mainly children, show strong impairment and an altered immune response. No one's looking at that. An imbalance has also been demonstrated in the production of these cytokines, the language I just spoke to you about. And what are the anti-inflammatory cytokines produced by these white blood cells in the ASD patients? And these cytokine imbalances reflect changes in behavioral symptoms in the ASD subjects. All of this is in a well-published, peer-reviewed journal article, Vicina Salco. And I can send you the information on that article. Next slide, please. Well, let's look at some more references to prove exactly what I'm saying. I want to apply psilocybin to autism spectrum disorder. The first thing I need, remember, I need a proof of concept, a preclinical. So I have to take psilocybin and show that it rescues sociability deficits in an animal model. Now, there are good animal models. There's a valproic acid model of psilocybin, and we now know that a drug that's approved by the FDA can cause autism if you give it to pregnant ladies who are at certain stage of development of the fetus. Behavior in metabolic disorders, the increased production of acute phase proteins and cytokines, this inflammatory language, may promote and contribute to the development of the symptoms we're seeing in autism spectrum disorder. The microbiome. We now know that ASD children have a mix of gut microbes that is distinct from that in children without the condition. And all of these articles are referenced at the bottom. Next slide, please. All right, what I want to do now is thank you for allowing me to talk to you. And now I want to open up the questions and answers, because in my experience, lecturing only provides certain information. Most of the information comes from the questions and answers. Thank you, and I'll open up to questions.
Thank you, Dr. Hausman. We have one question already in. Guest 524 is asking, can you give references where science has shown that microbes travel along the vagus nerve?
Yeah, absolutely. It's called the Brock's hypothesis, B-R-A-A-K-S. And Brock postulated that the cytokines migrate up the vagus nerve into the brain. I was recently involved with Dr. Julia Peridiri, who's going to be speaking on a panel later today, in which we did a study with Michael J. Fox Foundation. So an animal model was created which had Parkinson's disease. They take the human gene, they put it into the brain, they transfect the DNA in the brain, and the animal develops Parkinson's symptoms. And Julia and I studied the inflammatory molecules in the substantia nigra, which is a specific area of the Parkinson's brain. And we actually showed modulation with a substance from mushrooms called ergo-D2, ergothioneine combined with vitamin D2 in a high level, that we can modulate genetic expression in the brain of the Parkinson's animal. I think that proves to some degree Brock's hypothesis.
Thank you. Larry Selkov is asking, are any of these drugs being used to treat opioid disorders?
Yes, yeah, absolutely. We now very interested, the receptors. That's an excellent question. We have to now look at the language for opioid addiction. And the language is not just in the brain. The language is throughout the body. The formula of life is the same. Whether you have PTSD or you have depression associated with cancer or autism or Alzheimer's disease, or even arthritis, even liver disease, NASH, which is non-alcoholic liver disease. The formula is the same, and it's the language that we have to go after. And I think opioid addiction is something we have to study. I think there are receptors for opioid addiction that would be counteracted by these anti-inflammatory molecules that are in psychedelic compounds, psychedelic mushrooms.
Thank you. Dr. Kellen Thomas is asking, what duration do you expect? Oh, sorry, escaped now. What duration do you expect a single dose of psilocybin anti-inflammatory effect to persist?
We're going to know that in 3 months. We have an ongoing study in Italy, and we're looking at 3 doses of psilocybin. We were able to produce, I'm not here to comment on my company, but I'm proud of it. We were recently able to produce greater than 95% pure synthetic, non-GMP, and that we were going to test it at 3 separate doses, and then we're going to look at the behavioral abnormalities in the valproic acid ASD rat, and then we're going to redose it, and then we're going to look at what time period in the development of autism. So you can look 9 days postnatally, 30 days, 60 days, and we're going to see, do we have to redose? No one's ever done this before, so we're very excited. Stay tuned. Hopefully we'll have announcements by January 2021. Thank you.
Thank you. Our Gaurav is asking, can you elaborate on the biomarkers you're trying to identify as indicators of psychedelic medicine therapeutic effects?
Yes, we know them. I'm working with a company called Thermo Fisher, which is one of the largest genetic companies in the world. We have a lab in Vienna, Austria, and we have identified the key let's say, the inflammatory cytokines that are on different disorders. Even heart attacks have their inflammatory markers. So we have identified 49 RNA molecules and 9 microRNA molecules. These are smaller molecules that the RNA then translates into microRNA. I'd love to tell you what they are, but we're planning on filing patents in the immediate future. So thank you, but when we publish our literature, we'll be able to show you what are the exact cytokines that go into inflammatory disorders, including COVID-19.
Okay, thank you. On that topic, Pamela Thompson is asking, are you saying that probiotics that are helpful? Are you saying probiotics are helpful in stopping the inflammation, and the psilocybin has some part in creating a healthy gut microbe as well?
Yes, I do it myself. I don't preach what I don't take. I don't preach wine and drink. I don't preach water and drink wine, okay? I believe in the microbiome. I take probiotics every day, and I want to prove that. And we're setting up now antibody studies. I'm consulting because I just got nominated to the board of New Generex Immuno-Oncology, which is we're developing a vaccine for COVID, and we're going to be looking at all of this. We're going to look at the microbiome, we're going to look at the cytokines, and we're going to see what happens in your body with antibody production. And we're going to look at different types of antibody, neutralizing antibody, IgG, IgM. The jury is still out on many disorders, and we have to have an open mind as we go into the 21st century. Well, we're in 2021. We're in the 21st century. Thank you.
Thank you. What is the optimal clinical approach to understand the gut biome fecal sample?
I'm sorry, say that again.
What is the optimal clinical approach to understand the gut microbiome fecal sample?
Absolutely, we're doing that now. So what we're doing is taking fecal samples. You know, there's a big promotion now in medicine that you should. I don't want to talk about that. It sounds nauseating, but to eat your own feces. And they're saying that we should now treating disorders by taking feces from people who have recovered from those disorders and use them. I don't want to look at that. What we're doing is, for example, in the experiment in the autistic rat, we're looking at fecal specimens at different stages of development when they have autistic, 9 days, postnatally, 30, 60, and we're now going to look at the expression of the genes to identify these subspecies of microorganism. So in the next 2 or 3 months, we'll have a lot of data that we could tell the scientific community as to what exact species abnormalities are present in autistic spectrum disorder.
Thank you.
Just keep in mind that every bacteria has a different genetic code. So we can identify specific bacteria based on their genetic codes.
Wow. Good to know. Thank you. Do you believe the endocannabinoid system, CB receptors, but also TRPV, PAR, etc., is related to all of the dysfunctions that you're talking about here today?
I think so, but I have not had time to look into that. One of the areas we want to look at is NASH, which is a major problem. I'm a transplant surgeon. So liver transplants, kidney transplants, and we had a big program on NASH at Cedars-Sinai Medical Center and UCLA in Los Angeles. And so we were dealing with that because a lot of NASH patients need a liver transplant. And so I think those receptors are going to need to be identified. And I'm very interested in the near future being involved in NASH and identifying the receptors. I, though, want to look at psilocybin, but obviously the cannabinoid system probably has an effect there as well.
Thank you. Any idea of clinical trial about use of psychedelics for ADHD?
Yes, I mean, you know, ADHD is a common disorder. It's not a disease. Just like dementia is not a disease. Dementia is a symptom of Parkinson's disease. It's a symptom of Alzheimer's disease, a symptom. You can even be stressed and have dementia. And ADHD is the same thing. You can have attention deficit disorder in autism, attention deficit disorder in epilepsy, attention disorder in depression. So it's very common. And I think the same receptors are going to be, there's a broad spectrum, but I think all these psychedelics that we're looking at are going to have receptor activity in all of these disorders, including ADHD.
Thank you. I'm laughing on the next question. The guest 354 is asking, is that the reason why dogs eat poop?
Oh, good question. It sounds, oh, we're laughing. But yes, did you ever notice that a dog sometimes eats grass?
Yes, of course.
Yeah, I mean, we think that it's a sign that there's mineral deficiency. The animal, nature, remember what I said in my lecture, nature is perfect. We think we know. And it was, you know, as I think it was Socrates who said, you never know, oh no, it was Plato. Plato said, you never know the answer, you only approach the answer. So we've taken an animal, we've domesticated it, we think we know what food it should take. The animal is teaching us something. Why is the animal eating grass? Why is the animal eating its own poop? I think, and again, why is medicine now feeding poop from diseased people to normal people?
Wow. Thank you. Dr. Hyder is asking, and I might be mispronouncing this, NAFLD is cured by cannabinoid and capsicum combination drug therapies as it binds to the TRVP receptors,
right,
to suit and liver. So do you have any example of combination drug therapies?
No, as I said just a few minutes ago, I want to look at that. And we have an interesting model in NASH that we will be looking at with psilocybin. And then we're going to look not just at NASH by itself. Let me give you an example. Diabetes is a terrible disorder. But diabetes is really, type 1 and type 2 diabetes are inflammatory disorders. Yeah, type 1 may be more genetic, type 2, there's an overlap. We think now that the liver becomes insensitive to production of insulin, and that's due to the corrupted microbiome in the intestine. And NASH may be an outshoot of that because a certain percentages of NASH have diabetes. And so there's common receptor activity, and I'm very interested in looking at that in the immediate future, but I'm very focused. If you look at too many things, you get no answer. I want to take psilocybin, psilocin, maybe another psychedelic like baeocystin, which we're now producing. That's a very important derivative within psilocybin cubensis, and we want to combine that with psilocin and psilocybin and see if we have an entourage effect. And we also want to look at the receptors in NASH, and we'll look at the TRP as well. Good question. Thank you.
Thank you. Guest 654 is asking, it has been my observation that Parkinson's symptoms and amatoxin poisoning has near identical symptoms, just on very different timelines. Is there any exploration of a fungal cause?
A fungal cause? Well, we do know that fungal disorders have brain effects. They can, the disease can get into the brain through the blood-brain barrier. How it gets there, we don't know. Maybe through a vector that penetrates, because usually the blood-brain barrier protects you. I'm not sure how toxins get through the anaphylatoxins. For example, mushrooms do have toxins. How they will get through the blood-brain barrier. As I mentioned before in my lecture, dopamine doesn't cross the blood-brain barrier. Serotonin doesn't cross the blood-brain barrier, yet they're produced in high concentrations in the peripheral of the body, in the small intestine. So we have a lot of knowledge we have to explore in that area, but it is a good question.
Thank you. Guest 847 is asking, what are the prospects for a low to moderate dose of psychedelic to help people overcome shyness in social situations? And what would be psychedelic most likely to prove helpful?
Well, we have to, I don't know the answer. So, I tell you what I know and what I don't know. I haven't thought about that. I'm looking at more severe disorders that, the FDA and the regulatory authorities in Europe might get more excited about it. But obviously the receptor activity and social changes and social awareness overlap probably those in autism and other disorders. But I've never thought about it, but it is, it's a reasonable question and probably would have a definite effect with psilocybin and cannabinoids.
Thank you. Guest 354 is asking, all of this is just going over my head and it's quite frustrating. What would you recommend to a young mind seeking to understand all of this and eventually contribute to the narrative?
Oh, good. It's funny. I'm so trained, maybe I'm missing, what the average person needs to know. I'm so focused. That's a very good question. I think you have to, in answer to that, just like myself, I'm going to be 80 next year. You have to be your own advocate. Medicine is not your advocate. I'm talking from the heart now. Doctors are not your advocates. You only go to them when you're sick. You have to seek knowledge. And that knowledge is out there. So don't rely upon anyone just saying something to you. Seek justification of what you're hearing. And when you go to a doctor, ask questions. There are no such things as a stupid question. There are only stupid answers. So to answer to this gentleman, the answer is out there. Look at the published literature. Put the key word searches in. And again, I'm an open book. You have questions, come back to us. I'll answer them. Thank you.
Thank you. James Sandler is asking, if you're saying autism is a spectrum, could you extend this to say that all humans are somewhere on it? Wow.
I would have to probably say no, but it may be yes. We have to identify these subsets. Now, we know there's Rett syndrome, which is a genetic disorder that's part of ASD. We know that autistic children have epilepsy, so that's another subset. If we come back to the study that I showed you by Mindmen, Zynerga, Fragile X, a certain percentage of Fragile X is autistic disorders. Is it possible that the statistically significant part of their study, where the P was 0.02, is the Fragile X group that has autistic symptoms? And we need to identify the language that would differentiate between these subsets. It's my theory that the microbiome and the inflammatory molecules, cytokines, in conjunction with behavioral testing, will give us that answer. And that's what I'm working on.
That is beautiful. Thank you. Julian is asking, appreciate your willingness to share. Where can we contact you?
That may be Julia Peridiri. I'm happy to, I'm a chief medical officer for Nova Mentis, which is an up-and-coming company. If you Google me, you'll see how to contact me, my email, and I'll answer any questions. And I've said to Gaurav, I'd be happy to have a blog. I mean, if you can't answer a question, then you don't know, all right? You have to be able and able to answer questions. My email address is marvinshausman@gmail.com, Marvin, middle initial S for Stanley, Hausman @gmail.com, and I will always answer your question. And if you Google me, you'll see some of the work I've done. And if you talk to Gaurav and Patrick, they'll also tell you how to get in contact with me. And I'd love, if people are interested in what I have to say, to do a blog or a podcast for the organization. Thank you for asking that.
Thank you. Is there anything else you would like to add?
Wow. The one thing I'd like to end is the thing about advocacy. We have to take care of ourselves. Life is an experiment of one. Whether you have breast cancer, now my wife is now chairman in Florida of Susan G. Komen Breast Cancer Walk for the Pink. I see women who are devastated, families devastated. Disease is awful, whether it's breast cancer or autism or depression or PTSD. We know so little. I want to plead with people to become your own advocates. Life is an experiment of one. Psilocybin has stigma on it, but it could be very helpful. We have to overcome the history, which is positive, that the development, it's nature, it's perfect. Psilocybin is a food. You're eating it. It has things we don't know about. And we need to get the world to start listening to us. And I want to leave a legacy. I hope in the years that I have left that I can leave a legacy and show the application of psilocybin, psilocin, and other psychedelic tryptamine derivatives to cure this terrible, devastating disease called autism spectrum disorder. Thank you very much.
Thank you, Dr. Hausman.